I'd like to welcome you to our workshop on the topic of recombinant protein-based COVID-19 vaccines. We're very pleased to welcome select members of the industry to this workshop. And the goals of the workshop, as articulated on the website, are to provide a forum for product sponsors to discuss progress and technical challenges in the manufacturing when changing strain composition to currently circulating variants of SARS-CoV-2, and also be an open forum for collaborative discussions to facilitate advancement of recombinant protein-based COVID-19 vaccines. We will have occasional opportunities for Q&A, and I will help queue those up. And we have a very busy schedule. And so without any further ado, I'd like to welcome our first speaker of the day, a man who needs no introductions, the director of the Center for Biological Evaluations and Research, or CBER, here at the Food and Drug Administration, Dr. Peter Marks.
Thanks so much, John. So I'm mainly here just to welcome everyone and try to set the stage a little bit. First of all, thank you so much for taking the time today. I think it's been easy with all of the noise around mRNA vaccines to forget that protein-based vaccines represent a very important part of our armamentarium for vaccination. And spoiler alert, I'm happy to say that, I don't view any one vaccine technology as the be-all and end-all. I think that mRNA vaccines are great, viral vector vaccines are great, and protein-based vaccines are very, very important. And in fact, the potential flexibility that we could have in the ability to generate vaccines rapidly with protein-based vaccines, I don't believe has been fully exploited. And I think today is a chance for us to talk about that relative to COVID-19, but also may have implications for the larger field of using proteins for vaccination. So I'm really looking forward to a thorough discussion today. I think we need to think about this problem essentially freshly, and start to think about how we can address some of the limitations with the current systems that might be slowing things down from getting to market as quickly as other possibilities. Because, as I say, just to reiterate, I think that if we address some of those issues, we might have another series of vaccines in our armamentarium that could be made as quickly as, for instance, mRNA-based technologies that might be as or more acceptable to certain parts of the population, but even more important, that might be applicable to other infectious disease may come in the future. Because again, I don't believe that there's one size fits all for vaccines and pathogens. And there may be some pathogens that come our way where protein-based vaccines will be key in addressing them. So really looking forward to the discussion today. Hope we can have a very frank, open dialogue, and that we try to take kind of a fresh look at what can be achieved here. Thanks very much.
Thank you, Dr. Marks. And as a matter of personal privilege, I want to thank you for all of your time and commitment to the federal government and your service to our country. Our next guest is my friend and colleague, Dr. Robert Johnson, with the Biomedical Advanced Research and Development Authority. Dr. Johnson is the director of the Medical Countermeasure Programs here at BARDA, and where he has responsibility for overseeing medical countermeasure development across the organization, with an emphasis on new technologies and partnerships to ensure sustainment of our current and future investments. Dr. Johnson.
Great, thanks so much, John. And Peter, thank you for those really nice opening remarks. And I'd like to start off by thanking all of today's speakers and panelists for taking the time out of your busy schedule to participate in this really important workshop. So why are we here, and what does this workshop have to do with BARDA? So if we go to the next slide, BARDA supports advanced development of vaccines, therapeutics, and diagnostics in support of the U.S. government's preparedness and response efforts to many different types of public health emergencies, including emerging infectious diseases. And as part of this overall effort and responsibility, BARDA has been supporting COVID vaccine development from the start. So if we go back in time about 3 years ago to January, February of 2020, we'll remember the initial US government-funded vaccine advanced development work really focused on rapid vaccine development, honing in on different approaches to expressing the S protein. We fast forward 3 years later and we look at the outcome of those really excellent public-private partnerships, and we see that we're in a very different state. So through these public-private partnerships, we've gone from no approved vaccines to having multiple authorized and licensed COVID-19 vaccines, hundreds of millions of doses administered, and are looking to move this vaccination into a rhythm similar to influenza vaccines, which are updated annually. And this is where we run into the challenge and BARDA's interest in this particular discussion in this workshop. So first, what is the challenge? We're thinking about this idea of going into a cycle similar to influenza vaccines, so it should be pretty well trodden ground at this point. However, there's a critical difference between influenza vaccines and what we have currently with the COVID-19 vaccines, which is all around the manufacturing platform. So as most of you know, the influenza vaccine strain selection is made in late winter to support a fall vaccine campaign. And that's largely driven by several parameters, but one of them is the manufacturing time that's needed. Manufacturing influenza vaccines include a variety of platforms, including recombinant-based protein vaccines. However, with the COVID-19 mRNA-based vaccines, we can have a strain selection cycle that is much shorter. And this is incredibly important, as the later in the year you can make the strain selection decision, the shorter the time between decision and start of vaccination, and therefore the more likely that the vaccine will match the circulating strain. This causes a challenge for recombinant protein vaccines to pivot as quickly as the mRNA vaccines can, and this is the issue and the challenge that we are looking to solve. So the decision isn't to move and adjust the decision date. Maybe that's potentially for some an option on the table, but from the BARDA perspective, really not the direction we're looking to go. What we're really interested in is the solution which involves accelerating the manufacturing process so that they can meet this shorter production timeline that we see with mRNA. As Peter mentioned, this could have wide-ranging implications, not just for COVID-19, but for influenza, as well as for other products. So some of you might ask, how do we do that? Frankly, if we knew, we wouldn't be having this if I knew, we wouldn't be having this conversation. We know this is an incredibly hard issue, and that's why we've brought together some of the leading experts in the field who have been working this problem, not just in the context of COVID-19 vaccine, but well before. And that's really the key for this workshop. We're not here to talk about a particular COVID vaccine. Okay? We're here to talk about approaches, challenges, and potential solutions to expedite strain change with different recombinant protein-based platforms. This could include items such as manufacturers taking a little bit of additional risk earlier on in the process, or incorporating new technologies such as better expression systems or ways to make seed banks faster. This effort to expedite strain change capability and the overall development process for recombinant vaccines is critical to retain recombinant-based COVID-19 vaccines as viable options, which closely aligns with BARDA's preparedness and response interests. 3 areas that are of particular interest are speed and quality of manufacturing, access, and looking to the future. So as we think about the need for the annual update, obviously manufacturing speed is important. I've already covered why that is and what we need to try, the challenges that we need to try to solve. But I just want to emphasize that it cannot come at the expense of quality. When we think about our commercial products that are being made available, quality always has to be a top priority. So when we think about these integrating new technologies, for example, or new platforms, we can't forget that quality aspect. Second, a critical part of a complete response is ensuring access. And to enable the broadest possible uptake and protection, as Peter emphasized, options are critical. Only having one option or one platform can leave some people without access, such as those with allergies, but it also results in a potential for greater risk if something were to happen to one of those platforms. So having options is really critical for having a good response and making sure that we can reach as broad a population as possible. Third, being forward-leaning. Peter, I think, did an excellent job covering the fact that what we're talking about here is in the context of COVID, but these solutions that we're looking at can have much broader implications. So for example, just thinking about the potential around Project NextGen. If you flip forward to one slide, please. Next slide, please. So when we think about Project NextGen, what are we really trying to do? And how does that tie back with our ongoing efforts? So Project NextGen has been indicated before, really focused on the next generation of vaccines and therapeutics. How do we improve on our current products. So such things as mucosal vaccines, particularly intranasal, and other approaches that may have an ability to improve transmission blocking capabilities, vaccines that provide a broader or longer duration of protection relative to current products, or broader pan-coronavirus vaccines. As well as included in that is looking at new, more durable monoclonal antibodies, as well as, and something that I'll touch on a little bit more later, advanced development of new technologies that will improve access and enable faster, cheaper, rapid, and flexible production of vaccines and therapeutics. And this could well include technologies that could fill gaps we are identifying in this workshop. So briefly touching on how these things kind of start to come together a little bit. When we think about how we do this, information from forums such as this really will drive as we think about how to partner under Project NextGen under some of our other efforts. So whether it's looking at some of the activities that will happen with NIAID, partnering with developers around phase 1 clinical trials, or with BARDA's efforts or with our public-private partnerships, looking at doing phase 2/3 clinical trials with monoclonal antibody developers, as well as the next generation of vaccine developers, or supporting moving new technologies and new products into the clinic, or also integrating them into GMP manufacturing processes. So when we think about the current, we're thinking about the future at the same time. And what is the common theme on all of these things that I've touched on? It's really the importance and the prominence of recombinant-based products in this area, whether it's moving them into the phase 1, as I mentioned before, or thinking about late-stage development, or in the situation that we have with the current vaccine products that are authorized for use. Therefore, just in conclusion, our shared task today, again, is not to focus on a particular vaccine and a particular approach. It's really to understand the technical and other barriers to rapidly switching between strains. Focusing on recombinant proteins is an initial point of interest. We need to understand the critical path and those activities that are the lowest hanging fruit in terms of being implementable without impacting product quality.
I'm pleased to welcome our next product sponsor, that is Dyadic. Mark Emalfarb is the president, CEO, and founder of Dyadic, and is joined us online. Mark, please turn your camera on, and we can't wait to hear from you. Thank you so much for joining us today.
All right, thank you very much for having me. I want to thank the FDA and BARDA for the invitation to this opportunity. So Dyadic is both a product and platform sponsor. So our platform is available to license out to anybody, whether it's academics, governmental agencies, or industry. So Dyadic is working in collaboration with a growing number of scientists globally to overcome the challenges the world is facing in being able to rapidly develop highly productive cell lines to address the needs for mass production of recombinant protein vaccines to combat COVID-19 and, of course, pandemic readiness for the future. Next slide. Over the past 3 decades, we've invested significant resources in developing and scaling our microbial filamentous fungal protein production platforms for both industrial and biopharmaceutical uses. Our history here is useful in that it meets the needs of a global population. For vaccine manufacturing must have speed, productivity, sustainability, and affordability for both developed and emerging countries. And Dyadic's novel C1 protein production platform, while initially developed for manufacturing large quantities of low-cost industrial enzymes and scaled up to 500,000 liters, has now been further engineered for pharmaceutical use, the rapid development and efficient yet economical production of large quantities of vaccine antigens and biologic drugs, such as monoclonal antibodies. During our transition to biopharmaceuticals in 2009, the C1 production platform received US FDA GRAS certification, demonstrating safety for use in human and animal food and feed. Additional safety and efficacy has been demonstrated for multiple vaccine and antibody products, including non-human animal primates. And in February, we completed dosing for a first-in-human phase 1 study for a C1-produced protein for a COVID-19 booster vaccine in South Africa, with additional human trials expected in India with our pharmaceutical partner. Next slide. So over the last 30 years, we've developed an advanced genetic toolkit, which enables us to rapidly develop stable cell lines that can routinely express high amounts of complex protein antigens at very low cost and very large volumes. After several rounds of random mutagenesis and genetic modifications, we generated a novel C1 cell line with unique morphology that led to growth under very low viscosity and high yield that can be used to manufacture antigens, antibodies, other enzymes and proteins at very large scale. Also, due to its novel morphology, C1 cells don't sporulate. Unlike traditional filamentous fungal cells, C1 cells have been and are being used under cGMP manufacturing conditions. Next slide. In order to improve the stability profile of the C1 host production cell through systematically deleting 15 active proteases that, if not deleted, would degrade the proteins being expressed from the cells. These C1 protease deletion cells are now able to express high levels of stable secreted antigens, antibodies, and other proteins, as you will see in the upcoming slides. Next slide. As I pointed out earlier, shorter glycans may actually have potential benefits for immunogenicity and longer lasting protection. The native glycosylation structure of C1 is close to mammalian, but it also has shorter, similar properties to what the baculovirus cell short glycan structures. Yeast, which have hypermannoses, we don't have those issues and problems with the C1 cells. We've also humanized the C1 cells and have versions for human monoclonal antibody production, as well as vaccines with potentially better benefits. Slide 7. In the post-COVID world, speed of development is critically important. Production platforms must have the capability to rapidly generate stable cell lines, and as little as 6 weeks, we've been able to develop stable cell lines with C1 that are highly productive that can be used in cGMP manufacturing. We're working to reduce this timeline even further. This is critically important because when trying to respond to emerging variants of COVID or other infectious disease, such as H5N1 or the bird flu, achieving under 100 days to production and purification for recombinant protein vaccines and or antibodies is needed. And we feel, as a growing number of scientists globally, that C1 can deliver on that promise. Next slide. C1 is an ideal choice for developing and manufacturing recombinant protein vaccines and antibodies, and that it meets the needs for global demand and response from a development, speed, productivity, and cost standpoint. In addition, the manufacturing process, which I'll speak to in a moment, is simple, straightforward, and cost-effective enough to be adapted for recombinant protein vaccine production at flexible commercial scales throughout the world. Next slide. The C1 platform has produced proteins and antigens that can be used in animal models for all routes or in human models of administration. The C1 platform routinely produces antigens in unparalleled quantities. We've actually reached a record 10 grams per liter with a livestock vaccine recently, which shows the power of the C1 cells and the combination of our advanced genetic tools. Most importantly, that we meet the productivity and cost requirements that would promote vaccine equity across high and low income countries alike. We've leveraged our industrial experience, 3 decades over the past 6+ years, successfully adapted and applied C1 cells so they can be used for the development and production of recombinant protein vaccines and biologic drugs for animals and humans alike. Next slide. In the COVID pandemic, manufacturing capability and capacity were and are consistently being challenged. Our up and downstream processes are well-defined, simple and straightforward. Our platform uses low-cost, readily available media. It grows under a wide range of pH and temperature controls at flexible commercial scales, well beyond what would be needed for vaccine production globally. C1 cells can be grown in standard stainless steel or single-use microbial bioreactors, which are readily available globally. This leads to an affordable, sustainable production capability with minimal infrastructure support requirements in emerging countries. Next slide. I think time is of the essence in a pandemic. In the production of recombinant protein vaccines, speed and yield has traditionally been difficult, especially with the challenges of pandemic or even seasonal regional, let alone global demand. The C1 platform has a range of 6 to 9 days from the working cell bank to the end of the fermentation process, when you can start to purify the product. Most traditional eukaryotic cell lines, like insect and CHO cells, grow much slower and therefore need significantly longer time. Next slide. In the EU's Zoonosis Anticipation Preparative Initiative, ZAPI, 2 different antigens were used, Schmallenberg and Rift Valley fever. They were compared head-to-head with a variety of different cells, including insect cells or baculovirus. The C1 platform demonstrated several hundred times the productivity in less time. Another benefit, unlike insect cells and CHO cells, C1 has no viruses, or like E. coli, there are no endotoxins to be removed on the downstream. This also helps with manufacturing costs, as less steps are needed in downstream processing. This again translates to improved productivity and speed to release a product for recombinant protein vaccines for pandemics like COVID-19. Next slide. Now I'd like to focus on our vaccine programs. Dyadic has undergone the process to develop a monovalent recombinant protein vaccine that is currently in phase 1 as a booster for COVID-19. This vaccine is a RBD vaccine, similar to the ones that have been discussed previously by SK. Next slide. During the pandemic, we generated a stable cell line in approximately 7 weeks using our site-directed integration approach. However, as noted previously, our genetic toolbox enables us to use even more integration techniques. This gives us the ability to target specific loci and to rapidly replace the antigen with a different variant in cell to respond to an emerging threat. Next slide. Based on the guidance from leading European and Israeli coronavirus and MERS scientists at the start of the pandemic, we utilized a smaller SARS-CoV-2 RBD antigen for its ability to generate potent neutralizing antibodies. In collaboration in an FDA EU approved CDMO, we completed our cGMP process to produce clinical grade substance with 99% purity, on which we made cGMP drug product, which was formulated and released for use in our phase 1 clinical study. Next slide. The rabbit GLP tox study showed no adverse events, and antigenic stimulation persisted throughout the study, suggesting a major beneficial effect to vaccination without local or systemic adverse events. This was a 4-dose shot, 3-dose study. So as a booster, you can imagine that the safety we're seeing is very, very well and very good in humans. Next slide. Following the tox study, we prepared our regulatory submission package, which was approved and dosing was completed earlier this year for first-in-human phase 1 clinical trial for C1 produced protein vaccine. So far, the DYA100 vaccine candidate has been well tolerated with no serious adverse events observed. Ultimately, this has brought the C1 platform to an enhanced technology readiness level to produce commercial recombinant protein vaccines. Next slide. I'll now demonstrate several of our next generation vaccine candidates under development. Next slide. We've developed stable RBD C1 cell lines for all COVID variants from Wuhan to Omicron BA.5. This provided us and our collaborators with the ability to make recombinant protein mono or multivalent vaccines rapidly at high yields at low cost that are stable at 2 to 8 degrees Celsius standard refrigeration conditions to address specific needs with traditional recombinant protein-based COVID-19 vaccines. Next slide. In collaboration with Virovax, an NIH-sponsored adjuvant developer, and as mentioned earlier, adjuvants are a critical part of the component of a vaccine from a recombinant protein. We've run numerous animal studies with several different RBD and other antigens over the past 2 years. We recently expressed a next generation potential universal SARS-CoV-2 ferritin nanoparticle RBD antigen from C1, combined with Virovax's adjuvant, which is currently in animal trials, that is intended to produce broader and longer lasting neutralizing antibodies against future COVID-19 variants. We expect initial data from this study sometime next month. Next slide. In a collaboration with third party, we are evaluating an AI-based predictive modeling system to design multivariant versus multivalent vaccines. This would mean designing one RBD with multiple variants in one cell line rather than combining monovalent antigens in individual cell lines in a multivalent model. If successful, this approach has a potential ability, like the approach used with Virovax, just mentioned in the previous slide, to rapidly protect against emerging variants, maintaining one cell line, producing a single RBD antigen or potentially full spike antigen from C1 at high yields at low cost very rapidly. Initial animal studies result demonstrating binding inhibition as effective as homologous antibodies and shows potential promise for other viruses beyond COVID-19, such as influenza. Next slide. This slide demonstrates that C1 also has the capability to express large amounts of full spike protein at very high yields. We successfully expressed the full spike protein at 1 gram per liter in 5 days before optimization secreted. The binding curves of the C1 produced spike were consistent with the SARS-CoV-2 full spike produced in mammalian cells, leading to the potential for full spike versions of COVID-19 vaccines. Next slide. Subunit vaccines offer the potential for safe and widely used vaccines against a variety of infectious pathogens. However, scalable, cost-effective, and rapid production of recombinant protein antigens represents a major challenge for global subunit vaccine readiness. The C1 platform can help address this challenge by enabling high-yield production of complex protein antigens in a rapid, scalable, reproducible, and cost-effective manner. This has been demonstrated by the expression of, among others, ferritin RBD nanoparticle antigens, including Wuhan and Omicron, with the Wuhan ferritin nanoparticle antigen produced at 3.5 grams per liter in 5 days. Next slide. We're able to achieve expression of the ferritin nanoparticle in a single fermentation, rather than in many other approaches where it's necessary to produce the antigen and the nanoparticle separately to combine it later. An additional step, making the manufacturing process more complex, longer, and costly. Next slide. In a rabbit study, following intramuscular injection of the C1-produced Wuhan GRBD ferritin nanoparticle vaccine with the Virovax adjuvant, the vaccine produced extremely high neutralizing antibodies after prime and boost. If you look at the antibody levels on this slide, there are over 100,000 antibody levels. That's an amazing difference between what we've seen in the past. Next slide. Interesting, the adjuvanted Wuhan GRBD ferritin nanoparticle vaccine administered intramuscularly demonstrated some effectiveness against the XBB 1.5 variant after prime and after boost, although not to the extent as the Delta challenge. Next slide.
Dr. Emalfarb, we are running a little short on time and people seem eager to inquire. So you might consider wrapping up relatively soon. Thank you so much.
Okay. This is an intranasal approach that showed not only did we produce high levels of neutralizing antibodies, but in the serum, the blood, it still showed neutralizing antibodies despite being administered in the intranasal. Next slide. I'll just skip real quickly. This is just another approach of in this particular case, a fusion protein that elevated the neutralizing antibodies. Next slide. This is just, again, if you want to mix it with influenza, we have data showing that we can make significantly more faster, quicker, and using less protein to get better results. And we just received a US patent note of allowance for our influenza C1 cells. Next slide. This just shows you that we've produced virtually every type of antigen and antibody that you might want to look at. Next slide. So beyond vaccines, we can also use the platform, of course, to make monoclonal antibodies, therapeutic proteins, antigens, antibodies for diagnostics, recombinant human albumin, and even enzymes for mRNA and other complex vaccines. And the last slide, as we look to the future, we think beyond the vaccine design and realize that the cell lines used to produce recombinant vaccines, whether it be RBD or full spike, are increasingly important. C1 can do it quicker, faster, and reliably at much lower cost at flexible commercial scales globally. And I want to thank BARDA again and the FDA for the opportunity to speak today.
Thank you, Dr. Emalfarb. And we have very eager inquirers, so we would ask that they keep it short because we are a little over on time. But first up is Frank Arnold. Mr. Arnold, you care to inquire.
Quick question. Can you comment on the glycosylation profiles and protein folding, especially for full-length trimers or spike protein? Oh.
Yeah, unlike other fungi like yeast, C1 has a much broader, deeper genome. It's much larger, so it handles the disulfide bridges very well. We made the full spike protein actually with the VRC. So any of you that have reach into them, they can share that data with you. But I showed you where we made the full spike and it was virtually identical to the full spike made by mammalian cell. And on the glycosylation issue, we have multiple cell lines from even a short MAN3 to MAN3/9, which is what we use for infectious disease antigens, to fully human glyco patterns for monoclonal antibodies.
Thank you. And the final question is from Sujana Prasad Chikiani. I apologize if I mispronounced. Please inquire.
So what is the reason for selecting Virovax adjuvant? What is its advantage?
Testing the Virovax adjuvant, is that what you said?
What is the reason for selecting Virovax adjuvant?
Well, you know, AS03 wasn't available to us at the time of the pandemic, and we've tested a variety of different antigens, and the Virovax antigen was used a prior version of it in the Bharat Biotech vaccine very successfully to induce TLR7, TLR9 cellular activity as well. So we have a very close relationship with them. So we chose it because we thought it would work better. And they're an NIH-sponsored adjuvant in the NIH-sponsored program with Wolfgang Leitner and his team. And they've been very supportive, and it showed dramatically improved performance for recombinant protein vaccines, or at least the recombinant protein vaccines produced from C1 cells.
Thank you so much, Dr. Emalfarb, and thank you so much for the question.